My slides now.
So welcome everyone to our machine learning seminar. Today we'll have a pleasure to have a presentation from Christian Thygesen. I don't know if I pronounce it properly. He's an industrial PhD student with Evaxion and University of Copenhagen with a group of Professor Thomas Hammelrijk from his probabilistic programming group. And we had the opportunity to have a presentation of Thomas last week. And today we will have a continuation or next step that will be presented by Christian, which will be then followed in 1 week by another presentation by Ola Roning. Christian, the virtual floor is yours. Please go ahead, share your screen and start your presentation.
Perfect. Thank you very much for having me. I'm looking forward to showing you what I am working on along with Thomas and Ola at the University of Copenhagen. So I will give a talk today on a publication that we wrote last year called Efficient Generative Modeling of Protein Structure Fragments using a Deep Markov Model. And it was published at the International Conference of Machine Learning and also presented at the Probabilistic Programming Conference in 2021. I want to start out briefly by just telling you who we are. So Thomas last week gave a talk about the general problem and who his group is. But my project is actually a collaboration between Thomas's group at the University of Copenhagen and the Danish biotech company Evaxion Biotech, and we're funded by the Innovation Fund Denmark. So I am technically employed at a company in Denmark, but I get to work on all of the cool stuff at the university and do fun science and manage my own time, which is ideal to me. So Evaxion is a biotech company. They call themselves an AI immunology company, which is sort of a made-up word, but it does describe what they do very well. They use AI to treat people with immunotherapies. So Evaxion has 3 arms. There's a bacterial disease vaccine arm, which is Eden, and that's basically a neural network that can predict how immunogenic a protein will be, and then use that to design a vaccine. Then there's Pioneer, which is a patient-specific cancer immunotherapy. So in this platform, we use artificial intelligence to pick out the bits and pieces of a tumor that differ from the healthy genome, and then we can design a treatment that targets that those specific mutations. So hopefully we can kill off the tumor without killing the host. And then finally, there is the Raven platform, which is a vaccine platform for viral diseases. And that's mostly where my work is. So this is where we try to predict some protein structures and design vaccines using some of these predictions. Last week, Thomas gave you an introduction to the problem of protein structure prediction, but for the sake of clarity, I just want to rehash some of it so that it's clear for this talk. So a protein is basically a linear polymer of compounds called amino acids that are covalently bonded together to form this long chain of compounds. And I don't know, can you see my cursor?
Yes, we can see it.
Okay, perfect. So this extended chain of amino acids is called the primary structure, and or the amino acid sequence. And there are 20 different amino acids. They all share the same basic structure, so a nitrogen atom and 2 carbon atoms, and then each amino acid differs on the side chain, which we here just call R. But this side chain can look many different ways, giving the amino acids a host of different physiochemical properties. So the These NNC atoms, they form the backbone of the protein, and these bonds between the atoms can rotate. And these rotations are called dihedral angles. And these angles are actually what define the structure of the protein. So if you have a short stretch of amino acids that fold together, you can get different local motifs. And these motifs are called secondary structure, and there are three different classes of those. So that they are alpha helices, beta strands, or coils. And coils can look any which way. It's based more or less a random structure, very flexible structure at least. And then if you have the full extended amino acid chain that's folded with all these secondary structures together onto themselves, it forms this globular fold, which we call the tertiary structure. And that's the final structure of a protein, at least the final structure of a single chain protein. This is not the full story. So if you have more amino acid chains going together, then you have a quaternary structure. So that's where two tertiary structures form a larger complex protein structure. But for the sake of this talk, we will focus on going from the primary structure to the tertiary structure, and that's what we call protein structure prediction. So traditionally, before the recent breakthrough of AlphaFold, protein structure prediction was treated as a sampling problem. So you would do a Monte Carlo sampling of different conformations, and you would guide this by a physical energy function. But the problem is that if you want to sample your way through this entire chain of amino acids, then the outcome space is simply too large because there are so many degrees of freedom. And this is called Levinthal's paradox. And it says that suppose you have a polypeptide, so that's a protein or a chain of amino acids, that there are with 100 residues. So that's 100 amino acids. This chain will have 99 peptide bonds, and therefore it will have 198 different phi and psi bond angles in the backbone. If we simplify things a lot and say that each of these bond angles can only take one of three stable conformations, then the protein can misfold into a maximum of 3 to the power of 198 different conformations. And remember, a protein of 100 residues is not a very big protein. That's rather small, actually. So this space is simply too large that we can simply sample our way through it and get to the final protein structure. It's called Levinthal's paradox because paradoxically, if you take an extended chain of amino acid and you drop it into some water, then it folds to a globular protein in a matter of nano to milliseconds. So even though it should be impossible, it happens. So in the protein structure prediction software Rosetta, which is one of the biggest breakthroughs before AlphaFold, they try to reduce the amount of sampling they need to do. So they observed that there are thousands of proteins that are already where we already know the structure that's published, freely available to everyone. So what they do is they split the amino acid sequence that they want to sample from into overlapping fragments of 3 and 9 mer 3 and 9 mer fragments. So that means 3 or 9 amino acid residues at a time. And then they look in the known databases what known structures these small fragments can actually form. And then instead of sampling 1 amino acid residue at a time, they can then sample 3 or 9 amino acid residues at a time with known structures. So that should something that we know it can actually look like. So then you will eliminate a lot of the impossible structures that you would otherwise get if you just use standard Monte Carlo sampling, 1 amino acid residue at a time. The problem with this approach is that if you try to build fragment libraries using simply the sequence of the amino acids, then you are very biased towards picking the same structure for every single fragment. Or so let's say you have a amino acid residue, a fragment of 9 amino acids, and you want to see what it looks like in real life, and you just take all of the fragments where you know the structure, they will all look very similar because you're simply just biased in the way you actually do the crystallization. And we know that there are much more variants when it comes to these small fragments of proteins. So we wanted to try and address that. So we developed this method that we call Bayesian inference for fragments of protein structures. For short, it's just Bifrost, which is more fun to say. And it is the first deep probabilistic and generative model of local protein structure. And we base it on a Markov model, basically. So it is actually a deep Markov model. And for those who are not familiar with the deep Markov model, it is simply a hidden Markov model that's supercharged with neural networks. So instead of having linear transformations in the transition matrices, you get to have nonlinear transformations. We extended a bit on the standard deep Markov model by adding a bidirectional recurrent neural network that would process the amino acid sequence and give us a global view of the amino acid sequence at each time step. And then we try to infer the latent variables for each amino acid residues. So we pass each latent variable through a transition network that's implemented to look like a gated recurrent unit, where we treat the previous latent state as the memory state, and then the hidden state from the bidirectional neural network as the update state. And then we pass these latent variables to what we call the emitter neural network. So that's the network that parameterizes the distribution over the dihedral angles. Since dihedral angles are periodic values, here we chose to model them as a wrapped student t distribution. So basically a student t distribution on a circle. It's important to note here that there is an explicit conditioning on the amino acid sequence. So we don't actually model the sequence, but we give the neural networks the amino acid sequence explicitly, which is smart because that allows us to sample from the model super efficiently. But I will get to that later. In order to estimate the model, we use standard stochastic variational inference, and that will require us to introduce what we call a guide. And the guide is simply a variational distribution that takes the amino acid sequence, and here we actually also give it the observed angles, and then let that infer the latent states. And then it's basically the same structure as the model, so it's a Markov chain and with a bidirectional recurrent neural network as well. And this parameterizes the distribution over the latent states in the model. Oh, and of course, the latent states are assumed to be standard Gaussians. So we trained the model on data from 3,733 protein structures that are freely available in the protein data bank. These were redundancy filtered, of course. So we only included high resolution structures. So that means only structures where we are fairly certain that each single coordinate is correct. And then we removed all redundant sequences in terms of sequence identity. So there will be no two sequences in the training set that are more than 20% similar. We split these sequences into overlapping 9-mer fragments, and then finally we filter out what we call disallowed angle pairs. And we can do this because we know empirically what dihedral angles behave like. And that's shown in the plot to the right, which is called the Ramachandran plot. This is the distribution that we hope we can model. As you can see, there are many different clusters here, but there are also some very sparsely populated regions. And this is simply because amino acid residues cannot form backbone angles that land in these areas. So here in the middle, for example, that's simply not physically possible. We do see some dots landing here in the middle, and that's an artifact of the crystallization process. So in order to get rid of these disallowed regions, we simply just remove every single fragment that has a angle pair located here in the middle of this plot. And the clusters here in the plot, they actually correspond to the secondary structure classes that I showed in a few slides previous. So we have the alpha helices here, a very densely populated region in the Ramachandran plot, and we have the beta strands over here, and everything else is actually considered a coil.
Sorry, Christian, these angles are given like relative angles or just in the global coordinate system?
So these are the dihedral angles of the backbone. So let's see, yeah, here. So between the carbon atoms, the nitrogen and the carbon atoms, we have some rotations around the bonds. And these are the angles that we show here. So this is the phi angle and this is the psi angle. Was that your question, or?
I mean, with respect to, I mean, what is the starting point of the angle, whether they have a common coordinate system or?
So this is simply just plotting the phi angle against the psi angle for all of the proteins that we include in our data set. So this is the general distribution of all protein structures that look like this.
Okay.
Perfect. So we trained the model using stochastic variational inference, and we evaluated the model on a holdout set and showed that the model distribution, which is here on the right, almost perfectly resembles the observed distribution. We do see some added noise as the model cannot learn perfectly that this middle region is disallowed. In order to see if it actually learned the behavior of every single amino acid, we wanted to show the per amino acid residue behavior. So if you recall, there are 20 different amino acid residues. Most of them look the same, but there are 2 amino acid residues that behave differently. So these are glycine and proline. And that's simply because of the structure of their side chain. So glycine simply has a hydrogen atom as a side chain, whereas proline's side chain is linked back to the backbone in a different place. So that's very rigid, whereas the glycine will be super flexible. And then we included leucine here to represent the rest of the 18 amino acid residues that all behave more or less in the same way. And we show that the modeled behavior in the bottom is almost identical to the observed behavior. So this shows that the model has not only learned the general behavior of amino acids, but it can also distinguish between the behavior of every single amino acid. Finally, we tried to evaluate the model's ability to capture sequence dependency. So one thing is that's fine, we can see how amino acids behave, but what if we chain them together because they affect each other? And so this is what we did here. So all of the yellow lines that you see hiding behind all of the blue ones, that's an observed structure. And all of the thin blue lines are samples from our model, conditioned on the sequence from the observed structure. And we see that if we take a structure that is shown to be alpha helical, then the model is very good at estimating or sampling from sampling structures that resemble this, and that's simply because we have a lot of alpha helices in the Ramachandran plot. If we take one that's a beta strand, it is more likely to sample something that looks like a beta strand, but it will still sample something that looks like an alpha helix. And finally, if we give it a coiled fragment, then the model has no idea what to do. And this is simply because if it knows that these amino acid residues together form a coil sequence, then it will be super flexible. And there will be basically no signal that says this should look in a specific way other than it's flexible. We compared it to the current state of the art for producing these fragment libraries, and that's of course part of the Rosetta software prediction library. And we get more or less the same performance as Rosetta does. Sometimes we are a bit better, sometimes we are a bit worse, but in general, it's the same performance across the boards. However, with our model, we have improved the runtime significantly. So this is a plot of the runtime against the sequence length of the target protein that you want to produce a fragment library for, and we are 2 orders of magnitudes faster than the existing software. So that's very encouraging. So now that we have this model that can produce fragment libraries, basically we have a model that can produce that can sample from the local structure of the protein and give us an explicit likelihood. What are the next steps? Well, we've observed that simply focusing on angles is not enough. And I've tried to visualize that here with these cartoon representations of protein structures. So in the top row, you see a protein structure where I've introduced an error in one dihedral angle of half a radian. So basically, notice that if you introduce an error of half a radian, then the structure in general will look, the error from this error, the error from this angle will propagate out, so the coordinates of the protein structure will look dramatically different, even though it's a rather small error in one point of the amino acid chain. And if you superimpose these structures, they will sometimes look very similar, even though you have just a single error, but often they will look very different. And this is a problem, because this will imagine that you have small errors in every single dihedral angles of the backbone, then you will get a completely different protein. So we will try to address this using by modeling the 3D coordinates as well. This is a very simple graphical representation of the model that I've shown before. So it's a Deep Markov model again, but with a recurrency between the latent variables. And then from the latent variables, we get the parameterized distributions over phi and psi. And then we actually want to take the mean values for phi and for psi and reproduce the 3D coordinates. And we can do this using a known algorithm that's deterministic. We can then take these coordinates and apply a model that was developed in Thomas's group previously called Theseus PP, probabilistic programming. Well, they implemented a known algorithm in a probabilistic programming language. And this model is a model of protein superposition. So that will give us a likelihood that 2 structures or 2 sets of protein coordinates will originate from the same latent mean structure. We do this by sampling a unit quaternion, constructing a rotation matrix, and then applying it to our sampled or predicted coordinates, and then comparing this to the observed coordinates over here. And if we roll this out and make a very complicated flowchart about how things are going to look, then it's going to be it's going to look similar to what you saw before, but now we just have this p-nerve algorithm that transforms dihedral angles into coordinates, and then we can simply compare these using Theseus to the observed coordinates, and this will give us an explicit likelihood over 3D coordinates that we can combine with our error and likelihood over the angles. So these are some quite encouraging results for me that came in yesterday and this morning. So we took this approach and we trained a model on a known structural domain. So and show that we can actually go up to pretty large or at least larger structures. So this is a 37 amino acid structure, so that's bigger than the model has previously been able to do. And this is trained with the Theseus error model. And over here, we get the results when we train it without the Theseus error model. And basically, we get a much better globular fold when we train the model using the Theseus than if we don't. The model basically has no idea what to do over here to the right. So in conclusion, this was the first deep probabilistic and generative model of local protein structure. We show that it has on-par performance with the fragment picker from Rosetta, despite using less input. And we have a much improved runtime. And finally, we have some promising results for extending this to small or even medium-sized proteins. And of course, thank you to my supervisor Thomas at the university and Anders and Christian at Evaxion Biotech.
Thank you so much, Christian. So let's start the discussion. Again, whoever would like to talk, just switch on your mic and just start talking.
So I will go first then. First, thanks a lot for a very interesting presentation. Second, you have a very impressive photo there of Thomas. He really looks amazing on that one. He should use that for everything. He should use that for everything that he has.
I have a much better one of him, but he told me not to use it.
Too bad. Then I was wondering, of course, as the directional guy, is that so what motivated your choice for a circular wrapped student T distribution on the circle?
Yes. So yeah, so we're over here.
Why
well,
that
first
one?
Yeah. So obviously angles are periodic values, right? So we need something to fall between pi and minus pi, or minus pi and pi.
Yeah.
And the obvious choice would be the von Mises distribution.
Mm-hmm.
But for whatever reason, which I cannot seem to figure out, whenever I train a model using the von Mises distribution, it collapses. And I have honestly no idea why this happens when we use just the simple von Mises distribution. I've been trying a lot to figure out what's going on, but it just simply does not work. So it might be something with the implementation in the Pyro language, or there might be something with the way it handles the borders between minus pi and pi. I simply don't know.
That's
What's interesting is that Ola actually implemented a bivariate von Mises distribution in Pyro, and this works perfectly.
That's strange. That is indeed really strange. Okay.
Yes.
And did you try out other choices then, or did you directly go to the wrapped student from the von Mises?
Well, we went to the wrapped student T initially because we thought it might be because of the thin tails of the von Mises distribution. So if it's a normal distribution on a circle, then we figured maybe that's the problem. So if we sample something that lands in a sparsely populated area on the circle, then it might simply just give us extremely poor likelihoods, right?
Okay. See, that was the rationale behind it. Okay.
That was the rationale for going with the wrap student T. We might as well have gone with a wrap normal distribution. I think that would have worked as well.
Mm-hmm. Okay, but might just be interesting to see if by using perhaps some more choices, perhaps also the Cato-Jones distribution or some others, then just to see if you can even get some better results. It's hard to say. I could imagine that, but if you look at it in terms of pure fitting on the circle, that might be the easiest to just see the comparison and then see if you get a better one. But I understand your rationale for using the wrapped student T. That makes a lot of sense. Thank you very much.
You're welcome.
Saurabh, you want to ask?
Yeah. Hi, Christian. Thanks for the nice talk. Can you go to slide 12? You talked about using the coordinates instead of the trihedral angles, and won't that increase the number of parameters for you? I did not understand how it actually helped.
Actually, it only increases the number of parameters very slightly, because the p-nerf algorithm, which is depicted here, that's actually an analytical formula. So there will be no parameters involved there, at least not neural network parameters. Of course, distribution-wise, yes, we will increase the number of parameters immensely. You will go from having two, so let's see, if you have per amino acid, so we have 2 parameters for the latent variable and 2 for the for each angle. then you would also have 9 coordinates for each amino acid. So yes, you would more than double the number of parameters that you would need to fit.
Okay. Yeah, maybe I need to read. So that's confused me because how it exactly it helped, but maybe it's because of the PNERF algorithm. Maybe. Yeah. Thanks,
Yeah,
Alan.
but I mean, if there's something specifically you want me to elaborate, I'm happy to do it. But yes, the PNERF algorithm is analytical or deterministic at least.
Okay.
And then basically we actually don't sample the coordinates because we just reconstruct them based on the dihedral angles.
Okay.
And then
Okay.
we can compare these to the observed structures with some variance, and we can either model this variance. So this is what's shown here with the U. So we can either model this variance, or we can fix it to a certain value, or we can predict the variance directly from the variance over the dihedral angles. And actually, the latter part is what gives us the best results.
Okay, thanks. And also one small doubt in slide 7, maybe I was just trying to analyze you talked about the variational inference there.
Yes.
Yeah, exactly. So you said G's are the guides, which are the approximate distributions, right? If I'm not wrong, that's what you.
Yeah. So the G is a bidirectional neural network that passes the amino acid sequence and the dihedral angles, and then gives that to a neural network that parameterizes the distributions over the latent variables.
Okay. Okay. Thanks. Okay. Thank you.
Maybe if I could have a question to this slide 12 again. Tell me if I got it wrong, but for me it seems like you just introduced these coordinates in order to be able to compare to the maybe observed results that are given with respect to coordinates. But for me it looks like the sensitivity of these positions with respect to the given set of angles is very high, like extremely high. Like if you change an angle at some place very tiny, then you can expect a tremendous change in the position somewhere else.
Yes, exactly. So there's a downstream effect.
And then if you compute the gradient in your backprop, when you try to train your neural network, then perhaps this gradient is actually this sensitivity, right? Yes. So I'm wondering how it affects the capabilities of your neural network to be properly trained. Do you need to apply any special techniques to let's say alleviate this problem, or maybe this problem is not very
no, you're right on the money.
You.
You're right on the money. So that is a problem. The thing about so what's important to remember or notice is that this PNERF algorithm is actually differentiable. So the gradient flows perfectly through it, and it will connect with all of the neural networks in the right way. Hopefully, theoretically, that's what we're going for, right? But since we have a lot of parameters here in the coordinates, there will be this likelihood will dominate over the likelihood over the angles. So the trick that we need to do is somehow scale this in a manner so it has an impact, but doesn't disrupt the training completely. Because if we just let this, the likelihood over the coordinates be utilized fully, then we don't learn anything. So we need to find this balance where it has an impact, but it doesn't impact too much. Did that make sense?
Yes, sure. But let's say you apply this variational inference technique, which somehow also can give you this, let's say, sort of uncertainty of your predictions together with the mean value. And so let's say another question is, can you use that to somehow assess the certainty or uncertainty of your given predictions? You shown this nice plot that for this alpha, or no, it was this helical one. You had quite good agreement, and so low sensitivity or low uncertainty, and for other structures you had a bigger one. So is it something that can be useful when assessing the usefulness of your predictions, whether they are correct or highly likely incorrect?
Yes, I mean you can get that directly from the distribution parameters you model, right? So so we get the mean and we also get the variance of each dihedral angle, and that can be directly translated to the variance over the or let's say the uncertainty over the coordinates. So if you have a high variance, then you have a high uncertainty, right? So you can directly infer the uncertainty, at least the aleatory uncertainty, based on this. Did that answer your question?
Yes, thank you. It's very nice. And maybe one tiny question, because probably some others would like to ask also, just from the physical standpoint, how these proteins behave in reality when you were referring to these dihedral angles. My question is, because there is let's say, all possibilities of alignments of these pairs with respect to one to another. And my question is whether it requires some energy to rotate this. I mean, how flexible the structure is to just freely rotate in reality, or maybe it requires some driving force to rotate. Is it something, let's say, well recognized? Probably it is, but for me it is not clear.
So it's something that depends very much on the. So let's for simplicity's sake take a full protein structure. It depends very much on the type of protein and the location of the protein. So let's say you have a protein that's embedded in a membrane, that will be completely static in every single thing that's embedded inside every single amino acid residue that's embedded inside the cell membrane. So that will be unable to move. But it does, typically these membrane proteins have protruding amino acid residues, chains that will then move around. And they will respond to a signal that will cause a motor function to open the protein up and maybe import something in through this membrane protein. So that's one example where an external signal can cause these these protein structures to change actually. So that's also a reason why we need to be a bit probabilistic about it, because the structures that we see in the public data sets are snapshots of what the proteins actually look like. But in reality, they breathe, so they will flex and they will rotate and translate and do every move around in every which way. If you have a protein in an aqueous environment, they will be super flexible, at least on the outskirts of the protein. So Thomas showed last week that all of the hydrophobic amino acid residues, they will tend to cluster in the center of the protein, but all of the hydrophilic ones on the outside, they will be able to flex as well. So they're super flexible and can interact with their environment. So yes, there's a lot of variance in the way the proteins actually look, even though what we see in the databases are static because they're snapshots.
And are you aware of any database that shows any possibilities of different structures for a given amino acid sequence, just to show what possible combinations can be observed?
Yes. So in these public databases, there's structures that are deposited. They come from an assorted assorted methods of obtaining the structure itself. So typically they will be crystal X-ray crystallography structures, and they will be super flexible. That would be one snapshot because you fixated the structure of the protein in a crystallized environment. Other approaches are like NMR, so that's nuclear magnetic resonance. They actually give you more of a trajectory of how the protein looks like. So if you take a structure that's obtained through NMR, you will get 50 to 100 different conformations of the same protein. They will look very similar, but you will see that they actually differ more in certain regions than in other regions. And then you can show, of course, these trajectories and how the proteins breathe when they are in an aqueous environment.
So is this something that you can use as an input to your probabilistic model, maybe?
Yes, definitely. You can input that, and that's actually a good idea to use this.
Okay, thank you so much. I'm turning myself into silence, giving the floor to others to ask questions. Okay, so I can see that the question part is over, I think.
Yes.
And so probably you might be contacted by some people in the future, especially that the presentation has been recorded and will be available online. Thank you so much, Christian, for your very nice and informative talk.
Thank you for having me.
And we are waiting for the next week presentation of Ola. Have a good day to everybody, and to you in particular, and see you next week.
Cool. See ya.